Good morning children, I am Siddharth. Now I am going to teach you the structure of tRNA. tRNA is also known as soluble RNA. Here we can see what is tRNA. So tRNA is the transfer RNA. Transfer RNA. It is also known as soluble RNA or sRNA. Soluble RNA, that is sRNA. And this tRNA is single-stranded. It is only single-stranded structure, and it is the shape of tRNA is clover leaf shape. Clover leaf shape. But the three-dimensional tertiary depicts it as a inverted or compact inverted L-shaped structure. And here the third point is this tRNA acts as an adapter molecule in the translation process, and which can read the code on one end and on the other end which would bind to the amino acid, particular amino acid. So we can write it act as adapter molecule. Adapter molecule. And I will show you the structure. So this is the structure of tRNA, and the tRNA, the one end is 5' and the other end is 3', and the 3' is extended beyond the 5'. Only single-stranded structure. The one end is having 5' polarity and the other end is having 3' polarity, but the 3' end is extended beyond the 5' end. Okay, so here this clover leaf shaped tRNA consists of 5 arms or 5 loops. So which are the 5 arms or 5 loops we can see. So which are the 5 arms or 5 loops in the tRNA? The first arm is anticodon arm or anticodon loop. So here this is the anticodon loop or anticodon arm. Okay. So here also we can see the anticodon arm. So this is the anticodon or anticodon loop. Okay, and the second one, the second loop is T loop, and here this side is the D loop, and here this is the amino acid acceptor loop. Amino acid acceptor loop or arm. So these are the arms, and one more arm is there, that is the fifth one. This is the variable loop. Variable loop, and this variable loop, this is the variable loop, and this loop is the vestigial part. It has no role here. So this is the vestigial loop, and all other 4 arms or loops are having specific functions. And now we can see the first one, anticodon loop. What is the function of this anticodon loop? And this anticodon loop is the place where the anticodons are there. Suppose this is the mRNA. So mRNA, if AUG, this is the start codon that you know, initiator codon. So for binding this mRNA, this particular tRNA consists of an anticodon that is complementary to this AUG. So here this anticodon consists of UAC. So the anticodon UAC having tRNA can bind with the AUG of mRNA. So this tRNA is specific to a particular amino acid. So the tRNAs are specified for particular amino acid. Is it clear? So the tRNA, there is a starting tRNA, or there is a tRNA for initiation, and that tRNA is known as Initiator Trna. So that Initiator Trna must consist of UAC anticodon in the anticodon loop. Then only that can bind with the starting codon of mRNA. Is it clear? So this is the AUG is the start codon, and the anticodon means that is complementary to the start codon. So the tRNA, which consists of UAC as anticodon loop or anticodon arm, only can bind with the start codon of this mRNA. So this tRNA with the anticodon UAC is known as Initiator Trna. Okay, and there is no tRNA for stop codon. There is no tRNA for stop codon, and each tRNA is specified for particular amino acid. So this is about anticodon loop. And next we can see the T loop. And T loop is the amino acid, ribosome recognition site. That I write over here. So anticodon arm with the anticodons. Okay. And the second one, T loop. T loop is the ribosome recognition site. Ribosome recognition site. Here, in the T loop ribosomes, there are two subunits. Larger and smaller subunits of ribosomes are there in translation process for catalyzing the formation of peptide bond, and that that ribosomes are recognized by the T loop of this tRNA. Okay, and next we can see the D loop, the third D loop. D loop is the binding site of the enzyme amino acyl synthetase. This is the binding site of the enzyme amino acyl synthetase. Binding site is the D loop. And the fourth one, so here this the 3' and here the OH group is free, so this area is functional. This is the amino acid recognition site or amino acid acceptor arm. Okay, so this is the amino acid acceptor arm. That is the 3' or amino acid acceptor arm. Amino acid acceptor loop or arm for the recognition of amino acids. Okay, I'll show you. So here you can see the 5' and 3' end. Here the D loop, anticodon loop. So anticodon loop or arm, T loop, and amino acid acceptor loop, and D loop. And one more loop is there, that is variable loop. So this is the loop, but that is a vestigial part. It has no role. Okay. And next we can see one video.
The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA.
Okay, this is a way by which the tRNA binds with the codon of mRNA. Hope you understood everything about the tRNA structure. That's all for today.